We now welcome Rotimi Aluko to the stage. Rotimi Aluko, professor and Tier 1 Canada Research Chair, has pioneered research on the production and use of novel food proteins as nutritional tools to maintain human health and prevent diseases. His groundbreaking work has established the structural and functional basis to facilitate the development of food-derived peptides as potent health-promoting agents. In 2022, he received the American Oil Chemists Society Lifetime Achievement Award for protein and co-product research.
Thank you very much for the introduction. I think I'm very, really excited to be here, apart from the award, the honor to be a fellow of the Royal Society of Canada, but the opportunity to meet fellow scientists and social scientists is really very exciting. And my talk is going to be a little bit different, but it pivots into the previous talk in terms of mental health. Now this one looks at the, in terms of the actual human health. So very briefly, just to give you an introduction, because again, some of you, if you don't, if you don't understand what we mean by proteins and peptides, just to distinguish between the two of them. So usually proteins are very big polymers, of amino acids. They are very huge. They are present in our food products, all right? And then the peptides, we call them, they are more or less the small polymers of the amino acids, so they are very small. But the fascinating thing is that, once you take these big protein polymers and you digest them, you split them into these smaller peptides. For some reason, these smaller peptides become really very active that can actually have a very significant influence on human health. And so, usually, in these big proteins, we have some sequences that when they are present within the protein, because they are tied up, they are not active, but we can release them by simply doing enzymatic digestion. We sort of simulate nature, because when we eat foods, for us to benefit from the proteins in our foods, the enzymes in our digestive tract digest the proteins, and then the smaller peptides are then absorbed. But in this case now, since we're in the laboratory, we can adjust the type of enzyme we use, we can adjust the time, and so we have the opportunity to produce really a very, a wide variety of products that could have significant influence on human health. So once these peptides are liberated, they actually act as regulatory agents, and these regulatory agents can actually perform better than the original proteins. So some of these peptides, because again you have different types of peptides within the digest, it means that if you use the digest as a health aid, you can actually influence several biological functions. Okay, you can have some that can benefit one part of the physiological activity, one will benefit the other. So you can have more than one type of activity from these types of products. And so in general, when these peptides are released, okay, they provide, physiological benefits beyond basic nutrition, because normally when we eat food, when we eat proteins, it's supposed to give us amino acids to build our muscles so that we're very fit. But we're talking about additional benefits beyond that physiological, that provision of amino acids. And so very briefly, just to show you how we do this, is just a very simplistic representation of the whole idea. But if you have, basically, if you have, oops, I think I have to go back. So if you have a protein material, you will simply, add an enzyme, and then we digest for a certain period of time. And, when you do that, of course, not all the proteins are digested. So you have those that are digested into peptides, and then you have those that are not digested. And because the digested peptides are very small, they are very light, so they are not as heavy as the undigested proteins. So by simple centrifugation, you can actually separate the peptides from the undigested proteins. And so you can then take the soluble phase, which contains the digested peptides, and you simply freeze dry or spray dry them, and that constitutes what we call a protein hydrolysate, which now can serve as a health aid. And so just to give you an example of what we have done. So we did, in my previous slide, like I said, we did a digest of hemp seed protein. All right, hemp seed protein now is quite big, actually, in Canada, because we are the leading producers of hemp protein in the world. So what we did a simple digestion of hemp seed protein. And then we wanted to find out among people who have high blood pressure. So this was a clinical trial, a randomized clinical trial of people people who are hypertensive. And so we fed them 3 types of diets. So some people consumed milk protein, which is called casein, 50 grams divided into 25 grams each, twice a day. And then those that consumed the hemp seed protein that was not digested. And then we have the third group that consumed the hemp seed protein, but now with addition of the digested peptide, all right? And then what you can see here is the fact that trying to see whether this works. Okay, it doesn't work. Anyway, what you can see here is basically for the first group on the left that consumed milk protein, you can see the average blood pressure there is about 137 millimeter mercury. When we give them the hemp seed protein, the blood pressure goes down to about 532. This is over 6 weeks, by the way, so about 5 millimeter mercury reduction. And then, additionally, when you actually add the peptide, you can see further decrease in blood pressure to about 128. And this is highly significant because we know that a 3% reduction in blood pressure is known to be a very significant benefit in terms of reducing the adverse effects of hypertension. So this showed a very significant reduction in blood pressure after 6 weeks. So I wanted to look at some of the fundamental aspects of these results. Why did we get this? So what we did was to look at the level of toxic reactive oxygen and nitrogen species, because usually hypertension sometimes is a result of a vicious cycle where you have the high blood pressure induces the body to produce some very toxic free radicals. And then these free radicals damage some aspects of the cardiovascular system, and then that actually causes additional increase in high blood pressure. So it's a vicious cycle. So if you can reduce the blood pressure and also reduce the quantity of these very, very toxic free radicals, you might be able to suppress hypertension more effectively. So you can see here from left to right, you can see that, again, from left, the group that consume milk protein, and then when you give them the hemp seed protein, there's a very significant drop in the level of these toxic free radicals. And even when you add the peptide, you have additional decrease in the level of these toxic free radicals. And same thing, we looked at the peroxide. Peroxides are some of these toxic free radicals that are produced, okay, actually as we age or during hypertension. And again, same pattern, you can see that a progressive decline in the level of peroxide. These are very toxic compounds. Normally, our body will produce compounds to neutralize these toxic peroxides, but as we grow old, we lose the ability to sufficiently suppress some of these toxic free radicals. And so when you give somebody an exogenous source of these types of products, it can help to suppress the level of these toxic peroxides. And then we also looked at the ability of this treatment to actually tell the body to make more proteins, defensive proteins. So these are proteins or enzymes that actually help to defend our system against some of these toxic products, all right? One of these proteins is called superoxide dismutase, okay? The more you have this enzyme in your system, especially in the cardiovascular system, the better your body can actually suppress these toxic free radicals, because it's an enzyme that neutralizes these toxic free radicals. And here you can see, going from left to right, as you include the protein or even when you now add the peptides, you can see that there is very significant increase in the level of this beneficial enzyme. So, to summarize, because I now have a few seconds left, you can see that when we hydrolyze hemp seed protein, okay, it can actually lead to the production of peptides that have very strong antihypertensive and radical scavenging activity in the human subject. So this is in the human subject. It's not just in rats. This is something we tested in human people with hypertension. And apart from that, you can see that the hydrolyzed hemp seed protein reduce blood pressure, okay, and also enhance the plasma antioxidant capacity. This is the capacity of our system to combat these toxic free radicals. And the hydrolyzed hemp seed protein also reduce the plasma oxidative stress and the hypertension markers more efficiently than the unhydrolyzed protein. So efficiently, not only lowering blood pressure, but making sure that the factors that are responsible for the hypertension are also suppressed. Therefore, the general field of bioactive peptides provides a proven approach to convert regular big proteins, which usually are not very, very active, into smaller but more potent therapeutic compounds. And this hypertension project simply illustrates our ability to use some of these food-derived natural products to suppress these types of chronic disease. And this is very important because we know that most of the diseases are usually treated with drugs, but many people usually will cheat on their drugs because of the negative side effect. And it's the reason why some of these drugs usually are not very, very effective. But if we can substitute or at least supplement the drugs with some of these natural food products that do not have any negative side effect, then it might be possible for us to provide a better health care for the whole Canadian system. Thank you.